Well, hey there, everyone. Welcome back. You made it to about the halfway point for the circulatory system. Hopefully you got something out of the movie about blood, the two movies about blood, and the one about the heart. Later on, we'll be actually utilizing some of that knowledge and looking for some of those structures on an actual heart. So be looking for that. What we want to do now is we want to continue talking about the heart. This time we're going to talk about the physiology. So some of the things we're going to be looking at is the cardiac cycle, what's actually going on in your heart to get that rhythmic heart beating, what are some factors that influence heart rate and stuff like that, cardiac output, that sort of thing. I don't have anything else, so let's go ahead and get started. Okay, so when we're talking about the physiology of the heart, we're talking about how it works, okay? What causes it to beat and stuff like that. When we're talking about a heartbeat is a single cardiac contraction. Now, a cardiac contraction is going to be all of the chambers contracting. And they do this in series. The atria contract first, then the ventricles contract first. In order to make this happen, we've got two types of muscle cells that make this process happen. The first ones are autorhythmic cells, which control the pace of the heartbeat, coordinate the heartbeat so the whole muscle just doesn't spasm at one point. And then the contractile cells, which actually do the contraction, and that moves the blood, the squeezing part. The conducting system is specialized cardiac muscle cells. These cells initiate, one section initiates electrical impulses, the other one distributes the electrical impulses. This gives the heart its autorhythmicity, which means the cardiac muscle tissue can contract without outside stimulation. Your brain doesn't tell your heart to beat. It can influence how rapidly your heart beats, but your heart tells itself if it's going to beat. It doesn't have outside influences telling it that it has to beat. This diagram right here is showing us the conducting system. There's two parts of that. The first part are the pacemaker cells. These are the ones that are going to initiate how fast the contractions are going to take place. If you ever watch competitive rowing, like the Olympics or something like that, there's somebody at one end of the boat who's setting the pace. They tell the rowers when to row, okay? And so those are two nodes. One of them is the sinoatrial node, the other one is the atrioventricular node. We also have the conducting cells. So these are the ones that will basically transmit the impulse. These are the wires. Internodal pathways right here. We've got the AV bundle right here, which has branches into bundle branches, and then those go into Purkinje fibers down here at the end. And then as you can see, sections like right here and right here branch off, allowing that impulse to spread out to all of the contractile tissue of the heart. This is a diagram showing changes in the membrane potential of a pacemaker cell. These are in the sinoatrial node. And so it's important to note that this graph of the membrane potentials looks different than what we talked about with the nerve cells. One of the things, for example, is there's a gradual depolarization of the pacemaker cells, which we find right here. So there's not a stable resting potential like we do with nerve cells. Remember, we said with nerve cells, the resting potential is about 72, and that doesn't change. With heart cells, that gradually depolarizes before we get back up to that threshold potential. The two nodes will actually depolarize at different rates. Your SA node will depolarize, or the rate of spontaneous depolarization. So basically how many action potentials per minute. The SA node is actually faster. It's 60 to 100 action potentials per minute. The AV node is a lot slower, only 40 to 60 action potentials per second. So the SA node is going to set the pace. The AV node is going to sort of regulate that a little bit to allow contractions to spread throughout the larger sections of the heart. The SA node is going to depolarize first, and that's going to set up what's known as the sinus rhythm, or the bass rhythm for the rest of the heart. And that's it. We measure that, so that graph that you saw, we measure with a machine called an ECG or an EKG, an electrocardiogram. And so that's basically a recording of the electrical events of the heart. So we're monitoring those nerve impulses through that conducting system. And so we put electrodes, if you notice the diagram, if you notice the picture right here, we put electrodes in very specific places in order to monitor these movements. And so this is a very powerful tool to use because if we have abnormal patterns, those abnormal patterns are caused by abnormal impulse transmission, which leads to abnormal contractions. And so we can read that chart, and we're gonna break down one of these EKG graphs here in just a second. So we're gonna, based on what it looks like compared to what it's supposed to look like, we can diagnose different disorders. And you're gonna practice doing that with an activity that we're gonna do in class. So features of an EKG include a P wave. This is where the cells in the atria begin to depolarize. We have the QRS complex, which is depolarization of the ventricles. We have an R wave in here where the ventricles contract after that. We have a T wave, which is the repolarization of the ventricles. Time intervals involved. We have the PR interval, so the interval in between the P wave and the R wave. That's the start of atrial depolarization to the start of the QRS complex. And then after that, the QT interval, which is the time required for ventricles to undergo a single cycle of re and depolarization. And so here is basically what's going on in that conducting system during these contractions. So the first step, we have activity in the SA node, which activates the atrium, basically. That's going to send a stimulation through the tissues of the atrium until it gets to the AV node. And you can see that right here moving down. Now that takes about 50 milliseconds, so super duper fast. That gets us to the P wave, which we see right there. From there, it reaches the AV node. Now that stimulus gets delayed for up to 100 milliseconds. As you can see, the elapsed time has gone up by 100 milliseconds. This is going to signal the start of the atrial contraction, squeezing that blood from the atrium into the ventricles. From there, the nerve impulse is going to travel down the AV bundle to the left and right bundle branches. You can see in step number 4 right here. And then that is going to spread down and around the Purkinje fibers back up. Then the Purkinje fibers are going to send that impulse through the ventricular myocardium, so the myocardial layer of the ventricle. Atrial contraction at this point will be completed, and that nerve impulse will have, at this point, gone all the way through the ventricle tissue, so then ventricular contraction takes place. And so here we are right here with that S going into the R going into those S waves. There we go. Okay, so here's another breakdown of one wave, which is this section right here. So here is our P wave where the atria cells are depolarizing. The T wave is where the ventricle is depolarizing. And then we have our various intervals down here at the bottom. And so this section right here is one cycle where we go through this. Impulse cycle. Now, if we have disturbances to this particular rhythm, we have a lot of problems. And you may have heard some of these if you watch like documentaries and stuff like that. So one of them is bradycardia. That's abnormally slow heart rate. Tachycardia is abnormally fast heart rate, so if your heart is racing. We can actually have abnormal cells that create a high rate of action potentials. This is known as an ectopic pacemaker. These cells bypass the conducting system. Basically, they override it, which ends up disrupting the timing of the contractions. So these cells can cause a lot of problems and end up having to be removed. Here's some other irregularities that you can see, like premature atrial contractions. Various things like caffeine and stuff like that can basically cause the atrium to contract prematurely. That can disrupt the rhythm. We've also got our cardiac contractile cells. So these are the cells, we're kind of like halfway through what's actually happening. We've done the conducting cells to send the impulse all the way through. The contractile cells, that's going to form the bulk of the actual tissue of the walls of the cell. They're going to receive the stimulus from the Purkinje fiber. So once that impulse is spread through the Purkinje fibers, now the contractile cells are going to start working. Their resting membrane potential is a little bit lower. Ventricular cells, it's about -90. Atrial cells is about -80. Now, contractile cells have a very special structure on them known as intercalated discs. And these sort of knit cells together. They kind of act like the sarcolemma, the T-tubules, and skeletal muscles. They allow the impulse, that action potential to propagate in different directions, basically. So we have that sort of three-dimensional contraction process. And so they have various, a couple of structures that are in there. They run through the Z bands right here, for example. They've got these gap junctions that allow the impulse to travel through and into different cells and stuff like that. And so this basically allows the impulse to radiate outward instead of just traveling down muscle fiber strands like in skeletal fibers or skeletal muscles. So in cardiac contractile cells, the action potentials that take place are a little bit different than what in some of the other ones. They have rapid depolarization, as you can see right here, caused by rapid sodium entry into the cells. Then we have the plateau, which is here at step 2, which is going to cause by calcium entering into the cell from the interstitial fluids around the cell. And then we have repolarization right here, where potassium is going to be leaking out of slow potassium channels. And then another characteristic that's a little bit different, because there's still a refractory period, if you notice that's in there. We actually have 2 different refractory periods, okay? One of them is the absolute refractory period. That's that blue section right here, where cardiac cells cannot respond under any circumstances. In the relative refractory period, the sort of pinkish over there on the right-hand side, is when cells can respond, but only to strong stimuli. And then once we get past that, they can respond to regular stimuli, and so the contraction starts again. And so one of the things that's kind of interesting is the action potential happens first, and then there's a delayed reaction that takes place. So the action potential goes, and then we have the muscle contraction that happens after that, and then we're able to start this over again. And so basically, if you look at the different periods right here, the muscle contraction takes place during the refractory period of the action potential. So while the muscles are contracting, the conducting cells are resetting. And then when the conducting cells are resetting, the contractile, when the conducting cells are going through their action potential process, the contractile cells are resetting. And so we have this sort of ba-dump, sort of delayed reaction sort of thing that's going on, basically. Tell you what, I think that's kind of a clean break. It's a little bit shorter than how I usually cut these off, but I think it's a pretty clean break. So we'll go ahead and call it there. Go ahead and click on over right away in the actual cardiac cycle, where we're describing, now that we've looked at the contractions, or at least an overview of the contractile cells, now what we're going to do is look at the order that those cells contract to move the blood around, and then talk about like volume things and stuff like that. So if you have any questions about this particular section, let me know, send me a reminder, send me an email, ask me in class. Go ahead and click on over to the cardiac cycle video after this one. Otherwise, I look forward to seeing you all again soon.